So, hi, I'm Jeff Garnas. Very nice to meet some of you. I've seen many hours on Zoom screens and some of the other folks here in the audience that I haven't seen before, but um, it's great to be here. So I'm just going to give you a little bit of an update on what we've been doing in New Hampshire for the last 2 years, and I'm gonna tip my hat a little bit here. First mention that my background is very much not in the area of ticks or vector-borne diseases or anything. So I'm a little bit of a tourist here in this group, but I typically study the short version is the ecology and evolution and management of novel biotic threats in forest ecosystems. So I study things like the emerald ash borer and beech bark disease and beech leaf disease, and a new one, and a bunch of other forest pests is sort of the niche I've carved out for myself. So I guess I think of ticks in some ways as a novel pest in our area. We have, well, I'll show you some data showing that we knew that the Lone Star tick was around, but we did pick it up in our sampling, which is not common knowledge to a lot of people. And some of the other ticks are moving north and finding their way to the northern forest where they never were before. So I think this fits in. But I'll be honest that I am not a tick biologist by any stretch of the imagination. So this is just some of my lab group, and I realized I took a slide out that removed Casey Coop, who's been the leader of the field campaign for us for the last 2 years. He is a co-author on this little talk here, but we've got a sort of diverse group, and some of these people have helped out with some of the field sampling. And we learned how to do tick flagging. So I'd heard about tick drags for a long time, and then I realized that tick flagging was now the in vogue way to do this, and so we had to learn how to do it and how to make these really high-tech pieces of equipment. And then we learned that they're actually have their own challenges. So there's no great way to sample ticks, as it turns out. Who knew? So I grabbed a screenshot of this is actually my house up at the top there to represent one of the things that I thought was really interesting about some of the discussions we had early on was trying to think of, I'll say one of the things that attracted me to this group was the multi-state effort and the fact that people were trying to get their minds on the same page, even though they've been working across different disparate landscapes and sort of cultures and all sorts of things, and trying to produce a replicate data set across big areas seemed pretty appealing to me. And then the reality set in, and it turns out that that's actually pretty hard. And part of the reason that's hard is because our landscapes do differ quite a lot. So I don't know exactly what Rhode Island looks like, but this is a lot of what we see in my area is like this. So, homes with 5-10 acres of land and a lot of forest interface, a small yard relative to the overall land ownership. And so I would suggest, and my anecdotal experience is that, so I walk my dog through a trail, which you can't see here, back through the woods here to the Great Bay, which is over on the other screen probably, in New Hampshire, and that's where we get our ticks. So I mean, the yard for me is not that big of a deal in terms of tick exposure. It's when we walk through the woods and traipse through the higher vegetation and some of the invasive species. One of my favorite things to do when I take my dog back in these woods, I generally try to take out at least one Japanese barberry each trip. So about 100 years, I'll start getting a hold on that population. But anyway, so landscapes differ quite a lot. And I know that people are familiar with this, this approach, but I just I'm just going to show some of the data that we collected. It's new to me. It's probably kinds of things you've seen many times, but we collected in different zones, zone A being the yard itself, and zone B being the yard forest interface, and zone C into the forest a bit, which is, I guess, standard across a lot of the sampling that we did. And of course, this is how we sampled. And we were able to get 40 sites in southern New Hampshire, clustered in this southern New Hampshire area, which is Durham is right about there. So you can see they're clustered around where I live. I'd be interested in thinking more geographically widespread sampling, but that's of course logistically challenging, and recruiting properties was not so easy. So we did sample 40 sites in 23 and 24. Well, here's a little bit of a closer look. So you can see there's quite a gradient. Some sites we sampled only in one or the other year. The majority of sites we sampled in both years. We actually attempted in the second year to recruit properties from Bartlett Tree that had been treated so that we could do a comparison similar to what you've seen, but in the end that kind of fell through. And so we just have untreated properties for all our sample sites. And the other thing to note here is, like I said, so in this area and up north, it can be pretty rural, and it can be fairly large properties with lots of woods. When you get into Manchester and Concord, which looks like there's a sort of a non-randomness about the way that we sampled in 2024, but these are much more suburban, even urban kinds of landscapes, so very different. So I thought we got very low numbers, but seeing the numbers that on some of, I guess it's very variable. And we in 2023 in June and July collected, well, what's about 300 ticks total, and only about 62 ticks in 2024. And we thought maybe we were doing something wrong, but I think the answer is that's just the tick density at the sites that we were studying. And so that's what I'm going to show you. So I noticed that Nita and others used ticks per hour. We tried to equate that to some sort of distance. It really was a time metric that we used, but I'm reporting on ticks per 100 meters of flagging, and very, very low densities, but much higher in 2023, which fits the alternate year model for whatever that's worth. And overall, although I wouldn't say I didn't even do stats on this because I'm not sure we have the data to show it, but at least in 2023, Zone B, that forest edge, was the most had the highest tick density. In 2024, there were so few ticks that we didn't even really see that pattern. But that comports with, I think, other people's understanding of these systems. So I have a bunch of tables that are probably hard to read, and we don't need to really go into the details, but that's just another look at the same data. I was interested in what kinds of ticks that we got, and probably not surprisingly, it was dominated by the black-legged tick. Our sampling 83% in 2023 and 77% in 2024. I don't know if we were supposed to do this, but we collected all of them. This is a notable one, the Lone Star tick. I didn't know it was present in New Hampshire. It turns out it was not a first report, but the fact that we picked it up means it's got to be around. So I was very interested in the pathogen data that we got back from UMass. So this is really Juan's work more than ours, but I'm reporting it anyhow. So the only ticks that showed any positive pathogen detection were the black-legged ticks, and maybe everyone knows that too, but I didn't. And so in 2023, when we got the most ticks, the pathogen incidence was only 16%, so that seemed relatively low. It was quite a bit higher in 2024, but of course there are fewer ticks, so maybe that's just random stochasticity. 66% in 2024. And you can see the Borrelias pretty well represented in this group here, but we did get Babesia and Anaplasma, or plasma, I guess, or Anaplasmosis and Powassan virus and deer tick virus all showed up in our sampling, which suggests that there's cause for concern in New Hampshire as well as everywhere else. So not usually in the habit of giving time back to talks that I give, but I think I might this time. But so I just wanted to sort of end really with talking about some of the things that I've been thinking about going forward. I really enjoyed Bill's talk about the invasive species. I've read some of that literature. It seems really interesting to consider. So I would be interested in understanding how habitat and land use type influences tick abundances, given the variation that we have in our landscapes in New Hampshire and certainly in other states as well. I think it is important to evaluate the efficacy of chemical and other controls. I see that that's been a theme that we've talked about here. Feedbacks between ticks and invasives is also a really interesting thing to consider. And then I would be open to talking with any of you about things that we should be doing. Like I said, this is not my primary focus, but I think it's been a positive experience being connected with you guys, and we hope to continue it. So